So, hello everyone. Thank you for following our invitation to our hosted session, and I hope you enjoy. And I'm looking forward to a discussion and engagement afterwards. I'm Martin Rasch from Dynamic42, an organ-on-chip company in Germany. And today I would like to present our newest developments of real-time monitoring of tissues in organ-on-chip with an example of a gut-on-chip. So maybe a few words about our company. We provide organ-on-chip technology, education around it, training, and we are also a service provider. And yeah, you see a picture of our team. Who is doing the great work. We are located in central Germany, but we are about to step into the US during this year. So stay tuned and follow us if you like. And yeah, we combine a lot of scientific expertise in various fields with ease of use system. That is our vision. And we provide also lots of community support if you're interested. Let me tell you a few words why we are doing this. The overall challenges we have, not just in drug and therapy development, but also in biomedical research, life science, that it is dependent on high costs, lengthy processes in developments, and especially in the clinics, we have with drugs high failure rates. And this is in part partial depending on inadequate preclinical models we have currently. We see with animal models poor transferability, and also with static, classic, standard cell culture, that a single cell type cannot reflect a whole organ or a complex tissue. And the solution that we provide is organ-on-chip technology, where we can create advanced in vitro models for the preclinical phase and biomedical research. To reduce some of the drawbacks we currently have in the research and developmental pipeline. So for those who haven't heard of this technology yet, what is organ-on-chip? I've brought a general definition by the European Organ-on-Chip Society with me. I am board member there. And we talk in this space about a fit-for-purpose microfluidic-based device with different chambers and channels on it and compartments to set up living organ substructures that are vascularized. And this helps us to provide a controlled microenvironment. These chips we can, once the tissue has developed, attached also to a different kind of perfusion systems to simulate blood flow and nutrient supply as it is in the human body. And with this comes also biomechanical stimulation that alters the receptor decoration and the downstream signaling in the tissue and moves it towards a more in vivo-like, more physiological state. And with this, we try to recapitulate the dynamics and functionality of an organ. And with this, we want to improve the translation of data between in vitro and in vivo and contribute to close this gap. So when we talk about this, do you have to miss anything that you know from your research currently, especially from classical cell culture lab? And I have to say no. The D42 platform is highly flexible and you can perform all downstream analysis you know already. It is suitable to sample supernatants at any time point to check for biomarkers such as clinical parameters, cytokine profiling, or check for compound turnover or uptake depending on the organ of interest. You can easily recover the tissue from the chips, either as single cell suspension or lyse the tissue inside the chips for downstream analysis. But it is also possible to retrieve the whole tissue construct for immunofluorescence and histological stainings. Our platform is designed in microscopic slide format so that it easily fits into existing lab workflows. And you can do beautiful live cell imaging also. Here on this video, you can see a stained PBMC population in a liver model, and they attach and interact with the tissue over time and start to transmigrate. Live cell imaging is also suitable to investigate other parameters like reactive oxygen species formation or mitochondrial activity. And last but not least, you can check for enzyme activity, protein and transporter functionality. There are plenty of kits on the market available, and the protocols are all adaptable to our platform. And you can also use them to do your readouts. Now, how does it work? Let me show you a small workflow. It's pretty straightforward and contains the major steps you would do anyway in a cell culture lab, starting from sterilization over applying a coating for your cells that they need and desire, a cell seeding and growth phase until the tissue has fully developed. And then we usually start to perfuse the models. And after a small adjustment phase that can take up to 2 or 3 days, we usually do this over the weekend, where anyway no one wants to come to the lab. And then we can start our experiments and treatments, for instance, investigating therapeutic options. And after, for instance, 2 weeks, you can perform your readouts and downstream analysis that I presented you on the slide before. Yeah, and afterwards, cleanup as it is with every experiment, and then you can start the cycle again. And that's it. But, well, regarding the basics, it's not complicated, but there's more to discover, and that is what I want to present you today. Let's talk about real-time monitoring. I presented you readouts that you can perform along the way with sampling supernatants, or you do downstream analysis. But for those readouts, you have to manipulate the system, you have to touch it, and you cannot do it in really short intervals like seconds or minutes. Here, real-time monitoring is really helpful to really define the point of change in your experiment where you see an effect of what you are doing. And this is very helpful to monitor constantly vital parameters. It increases the model sensitivity. You can streamline your downstream analysis with this, and you can additionally use it as an internal quality control. And last but not least, it's there to make your research life more convenient and easier and straightforward. I want to present you 2 use cases on our gut-on-chip intestinal model. And before I start, I just briefly introduce this model and explain it to you. You see inside the chip on this schematic looks it as the following. This is the top bonding foil of the chip. This is the bottom, and everything in between is inside the chip. There's a porous membrane that compartmentalizes the chamber into a top and bottom channel. In the top channel, we set up a vasculature. And in the bottom channel, the organ-specific epithelial compartment, in this case, with intestinal cells. And when we start perfusing it, it also starts to outgrow beautifully in a villus-like structure as we find it in the human body. Now, in the intestine, there are many parameters that are of interest. One, for instance, is oxygen. With this, you can assess viability of the model, but you can also use oxygen to establish oxygen gradients inside the model, for instance, throughout the tissue, and monitor this. And this is helpful because a lot of people desire and talk about combining intestinal biology with microbiome. But we know that not all strains in the microbiota like the same conditions. Some are aerobic, some are anaerobic, and these conditions need to be modeled, and this is not easy, but control and monitoring of the oxygen can help to define the right conditions. And we can use it also to monitor the cellular behavior during therapeutic interventions. Now let me show you our sensor upgraded biochips. By the way, if you would like to see them in real life, you can also later on or tomorrow visit us at our booth 527, where we showcase them. So in the chips, we have integrated sensor spots in the channels. They are only 500 nanometer in diameter, and they are suitable for contactless measurement. They are, of course, biocompatible. This we assessed. And they are applicable to different sterilization methods like radiation or ethanol treatment, for instance. And we teamed up with Pyroscience, a befriended company, for a readout platform that we use to perform the contactless readout measurement. This is incubator and shaker compatible. It has an automated temperature and pressure compensation, and the readout occurs through transparent material in the chip at the sensor spots, and with this we ensure full flexibility, and this so-called fireplate can then also be put into the incubator to the organelle chip setup. Now here you see on the sketch where sensor spots are located in the top channel where we mimic the vasculature. We have currently one spot to monitor the oxygen consumption and level, and in the bottom compartment there are two in the inlet and outlet, and there you can also monitor changes when the medium is perfused through the organ, and this can also be helpful to establish lateral gradients that could be of interest, for instance, also in a liver model. We know that liver sonation is depending on lots of factors, and also oxygen plays a role there. And you see here, when we culture the chip statically, of course, not just the nutrients are consumed quite fast, also the oxygen level drops really fast on a very low level, implicating that the cells need better nutrition and oxygen supply. And when we perfuse it dynamically, we see quite nice differences between the inlet of the chip and then the outlet, and you see along the way the oxygen that is consumed by the tissue, and we see here also the recorded line for the vascular top. And and with this, this can be helpful to investigate different kind of questions. And of course, we did proof of concept also during the experiment to see if we can intervene with the oxygen consumption. We chose rotenone for this demonstration. It is an inhibitor of enzyme complex 1 of the respiratory chain, and yeah, we see that the administration of rotenone results in disruption of cellular consumption, oxygen consumption, in a concentration-dependent manner. Also, different kind of concentrations we applied, and you see, of course, lower concentrations have a more mild effect and a slower decrease of the consumption, and then you see that oxygen level increases again inside the model, and higher concentrations work much faster. And yeah, these the stop or the increase of the oxygen, of course, comes done by the constant perfusion of the model that brings in new oxygen, and partially also via diffusion through the biochip material. So to sum this up, the oxygen measurement is, of course, highly relevant to evaluate status of the tissue. It is an indicator of mitochondrial mechanism of action upon pharmacological intervention, and also an indicator of the energy metabolism in health and disease overall. And yeah, now we have a possibility to monitor this also in real time in organ-on-chip applications in our platform. And we've demonstrated a seamless non-invasive integration and also readout possibility that supports ease of use of this technology. And this was just the first use case I wanted to present you today. There is even more to get out of your experiment. We can also now monitor real-time tissue barriers. So the fundamental principle of organ-on-chip is the compartmentalization of tissue barriers under dynamic conditions. And this is an important regulator barriers in our body of the tissue microenvironment. So it controls passage of all kinds of molecules in our human system. And there are several vascular and also epithelial barriers in the body of high importance for homeostasis. For instance, very well-known barrier is the blood-brain barrier that restricts molecule transport into the brain. But we have also a gut epithelial barrier that helps us to spatially separate the luminally located microbes in the body to make us not sick, but to stay in the intestine and have a beneficial effect on our health. And these barriers can be monitored via a transepithelial electrical resistance, short TEER, and this is what we implemented as well in our chip platform. Here you see a schematic of the chip where we equipped our biochips with semi-transparent planar gold electrodes. And in this explosion view, you can see that the electrodes are positioned in the inside of the chip, of course, attached to the top and bottom bonding foil. And this enables you to get a readout throughout the tissue and check if the barrier is intact or not. And an additional circuit board that is attached to the chip enables the contacting of the electrodes and the signal transmission. And on the right side, you see real live pictures of them. And if you would like to see them in front of your eyes, just visit us. And that's not all. Also here, we laid a focus on ease of use and designed a measurement device for contactless readout, this time by ourselves. The measurement device can operate up to 3 chips. On each chip we have two models, so we can measure 6 models with 1 device in parallel. And the measurement interval can be adjusted from very short intervals in the second range up to 1 hour. And here you see an exemplary schematic how it can look like in the incubator. Signals are transmitted via Bluetooth to a computer with a data plotter software. And this is of course designed as an open platform, so open source data plotter software can be used for this. But we have tested one that is suitable for this, but also other softwares should work. And as TIR is temperature sensitive, it's a temperature sensitive technique, we thus avoid biasing the experiment by keeping the incubator closed at all times. Following, I would like to show you how it looks inside the biochip, and then also guide you through the workflow of the model setup and its impact on the TIR values. Here you see the tissue arrangement in the biochip. I presented already together with the electrodes attached to the bonding foils. And in between is the tissue located. And here is a real-life impression from an incubator setup. Now, during the setup of the model, we have a static culture phase where we seed in cells and let the tissue develop. And when the barrier starts to develop and tighten upon intestinal cell seeding, you see then also a major increase in the TIR values we get. And further, and very important, cell viability is not affected. So it is the same as it is in chips that do not have the TIR electrodes integrated. And yeah, you can see it here by ATP measurement of the tissue. And following, I'd like to show you how the system reacts to a treatment when we apply EDTA to the system. EDTA is known to disrupt tissue barriers, epithelial barriers, by capturing vital ions such as calcium. So we treated the model and saw immediate effects upon EDTA administration that the TIR values dropped down close to 0. And when we remove the EDTA, we can then also see that the tissue barrier recovers over time and monitor this. And we can also beautifully capture this via bright field imaging. So the electrodes are semi-transparent, so you still have the possibility on a bright field microscope to look at the tissue and to investigate it. You see control models, EDTA treated, and also after recovery time overnight. And in comparison, a standard technique is, for instance, Fitzy-Dextran-based measurement for barriers. And we see that this is not that sensitive to really detect significant changes and small changes in the tissue barrier. Of course, to complement this data, we stained for tissue barrier-related markers, such as E-caterine or ZO1, in the different conditions we investigated, and can see the effects also on histological, in histology. And you see here nicely the differences, especially upon EDTA treatment and then after recovery phase. So to sum this up, we demonstrated here the integration of semi-transparent gold electrodes, and the operation works via a simple plug-and-play workflow with flexible intervals for measurement. In our proof of concept study that we ran for 2 weeks, we could see the changes that appear when we administer treatment, in this case, for instance, with the EDTA. And the large electrodes, the large electrode area favors also the quantification and the normalization of tier values to make it more comparable also to other systems or applications. And the system shows high biological reproducibility. During several experiments we saw only minor changes in the tier levels that we recorded. Everything was pretty close together. And yeah, with this technology will be then starting in April, accessible for our fee-for-service partners, and later on also to the academic space throughout summer and this year.